Hello, everyone, and welcome to today's keynote presentation, Mapping the Inner World of the Cell, presented by Dr. Huang, Professor, Department of Pharmaceutical Chemistry, Biochemistry and Biophysics, University of California, San Francisco. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right of the presentation window and follow the process to obtain your credits. Now, please join me in welcoming Dr. Huang. I will turn the presentation over to him.
Hello, everyone. Here, I would like to share with everyone of our experience of getting to the inner world of the cell and try to see what's happening inside the cell. As all of us cell biologists want to do is that we really want to see all the details in the cell as to see what's happening, what's the mechanism behind it, and how we can use it to understand these fundamental processes and how we can use it to better our health care. And we know the cell is very small. It's just the fraction of millimeters, several micrometers there, and typically it takes a microscope to really get in there and see all these detailed structures. As all of us, when going to a not so familiar city, and we will need a map. We will need a map to know where everything is, where the most important attraction is, and so that when something happens, we know what it is, we know where to find it. So, and nowadays, with the advancement of the imaging techniques, we can easily take a picture of the city. Here, some of you may recognize that this is the city of San Francisco. We take a picture of the city from the satellite from well above the sky, and then if we have anything that's a good marker of the place of interest, we know where they are, we can draw a map of the city. So this is my own hand-drawn map of the San Francisco city with number of tourist attractions marked. Of course, one of the most important attractions that I suggest everyone to visit is UCSF and UCSF pretty much everywhere in the city. Actually, this is the similar experience is one to start drawing a map of the cell. That we have the cell there, and we first need to label the structure of interest. This is typically done by either fluorescent protein or fluorescent antibody, so that whatever structure of interest now has a color, now has some signal, some light signals that's coming out. And then we put our structure, put our cell under a microscope. And microscopes can capture the signal and produce an image. Then we send the image to our computer. The computer will analyze the image and tell us the answer. That completes the full path from cell to the cell. And this is basically involving a lot of the technologies the MindLab's been working hard to develop. And that's the pipeline that we are working on. And we've been working on structures in labeling, imaging, and analysis. And for today's talk, I'll mostly be focusing on the labeling aspects. And we've been looking at a lot of different cellular structures. We're using lot of different labeling methods. For example, we've been using antibody immunofluorescence in conjugation with superficial microscopy as a way to see the ultrastructures of the ciliary transition zone. That's a collaboration with German writer at UCSF. And we have also been developing methods that can label the DNA and using the CRISPR-Cas9 system so that we can follow the movements or the dynamics of genes in the nucleus in a living cell. More importantly, looking at all the other structures in the cell, especially for live imaging, one of the most commonly used practice will be the fluorescent protein. In this case, if we want to follow a protein of interest in a living cell, what we typically do is to make a fluorescent protein fusion construct. This protein of interest is fused to a fluorescent protein, and then we express this fusion construct in a cell. Then we get incorporated into the target structure, and then we can follow its movement, follow its dynamics, follow its life cycle. And this is so ubiquitous practiced, and it's extremely powerful technique as to learn what's going on in living cells. It's not to say that it doesn't have limitations. For example, here's a very nice illustration. We know the typical practice is that we have the target protein, we fuse it to the fluorescent protein we overexpressed in a cell, and that overexpression is changing the concentration or changing the expression level of this target protein. For many of the targets, this change is very well tolerated, so we don't see much thing going wrong. But for example, this is my own experience. There are certain proteins, if you just overexpress, things change. When we were trying to look at the endoplasmic reticulum, the ER protein, and we're trying to figure out the ER distribution of two proteins, one reticulum 4A, which is a ER tubule protein, and then when we overexpress it, the ER is full of tubes. And the other thing is the PIM63 is the ER sheet-forming protein, and when we overexpress that one, the entire ER morphology is changed. It's sheet all over the place. So it's just the overexpression is actually changing the structure of interest itself. And that makes it difficult to understand exactly what's going on with the endogenous ER, with the endogenous morphology of ER, with the endogenous expression of proteins. This is one task we're trying to go after. This is one method we're trying to establish is an effective way of enabling imaging and analyze endogenous proteins. Well, nowadays, with the advancement of gene editing techniques, it has been getting more and more routine to enable endogenous proteins using a green fluorescent protein GFP. And that is, well, you have a GFP gene, you add the homology arm with the CRISPR-Cas9 system, you can make a cut, double-strand break at the target site. Then homologous recombination will incorporate the GFP gene into the genome, and then endogenously this target protein is tagged with the green fluorescent protein so that you can track the dynamics of this endogenous protein. And this has been a very successful approach, and now we've seen many, many papers now published using the endogenous GFP enabling using gene editing. Well, it's effective doesn't mean that it doesn't have problems. The GFP gene is not small. It's over 700 base pair, and to make the homologous recombination efficient, typically people add like 1 kb homology arm on the two ends. So the whole donor DNA is more than 2 kb. It takes multiple steps of cloning to make that donor DNA, and exactly because the donor size, the insert size is big, the homologous recombination is not necessarily always efficient. So there are many cases of successful GFP integration, and that ends up with beautiful papers. There are also many cases, and some of them coming from our own experience, that it's difficult to get a GFP integrated cell line for certain genomic loci. So we're trying to change that. We know that in all this problem roots in the fact that GFP is big. Well, can we have some label that's smaller? It needs to be a label that's small, and it needs to be a label that becomes fluorescent by itself, so that we don't have to do a second step labeling. To solve this problem, the approach that we have coming up actually is the idea of from Daiichi Kamiyama, a former scientist of the lab, Sergei Sakini, a former postdoc in the lab, and together with Sui Feng, a extremely talented bioengineering graduate student in the lab, is that we don't have to label the target protein with the entire fluorescent protein. Instead, we can take the advantage of the split fluorescent protein system. In this case, we only need to put a small fragment of the fluorescent protein on a protein of interest, while we have the other big fragment just floating in the cytoplasm. And these two fragments, once seeing each other, they're going to complement and they're going to reconstitute a fully functional fluorescent protein. In this way, the insert can be much smaller. And for this purpose, unfortunately, Jeffrey Walden lab at North Marsman National Laboratory has already engineered a split GFP system. And the split point is between the 10th and 11th base strand. The two fragments by itself, especially the 10th fragment, which contain the residue that's going to form the fluorescent protein, they cannot mature by itself because a key catalytic residue is on the smaller fragment. And then only when the two fragments coming together, the fluorophore could mature and the whole thing could fluoresce. And even more importantly, the Walden lab has specifically engineered this split fluorescent protein construct so that they would very efficiently come together without the assistance of other protein-protein interactions. We know many of the split fluorescent protein constructs being engineered to detect protein-protein interactions. So you fuse one part to another protein, the other part to protein B. So if A interacts with B, the two split fragments would come together. And this is different. They just come together very efficiently by itself. And that's the system we're going to use. For some initial tests, we tried it to overexpress the GFP11 construct and GFP1 to 10 construct. You can see it works on a variety of proteins as a protein labeling tool. And we can also fuse GFP11 to a ER Newman protein and have the GFP1 to 10 tagged with the ER translocation signal peptide. So then sent to the ER, we can label ER nominal proteins or even extracellular domain of transmembrane proteins. So that's a general enough method to label overexpressed proteins. How about endogenous proteins? Well, the GFP11, in this case, is very small. It's only 16 amino acids. Including the linker, we can put everything into a DNA of 16 nucleotides, and we add 70 nucleotide homology arm on both ends. The whole donor DNA is just 200 nucleotides. You can directly buy it from DNA synthesis companies like IDT, and that makes not just the integration very efficient, but also the whole process cost effective, because we have synthetic donor DNA that just costs a few tens of dollars, and we don't need to do any cloning for the whole process. For the knocking, we've been collaborating with the Jonathan Weissman lab, partly inspired by the work done by Jonathan Weissman and Aaron O'Shea more than 10 years ago of making a fluorescent yeast library. So this library has a collection of more than 2,000 yeast strains, each strain with a protein endogenous tagged by the by GFP. So the important thing in this case is that it becomes a very powerful discovery tool. And usually people think fluorescent microscopy as something you know what to look at, then you can label it with a GFP. You can look at it, you can identify where it's coming from, you can analyze the behavior of the target protein. But once you have this library, fluorescent microscopy become a very powerful primary discovery tool because you don't have to take up a hypothesis. You can just go unbiasedly with an image-based screen and identify the gene of interest. And this yeast library has been very powerful in analyzing the abundance, substrate localization, and analyze the certain important pathways involved, for example, with yeast aging, a very powerful discovery tool. And now, with our method that we can tag endogenous proteins, we're wondering we can do the same thing for mammalian cells. So we've been collaborating with Jonathan Weissman lab, especially with Manuel Nanetti, a postdoc in Jonathan's lab, who's been working on CRISPR in gene editing. And the protocol that Manuel has developed was based on the Cas9 RNP method. Basically, we have in vitro transcribed guided RNA mixed with Cas9 and the nuclear protein complex mixed with our synthetic donor DNA. We do electroporation into the targeted cell line that's already expressing the 1 to 10 fragment. And then once the GFP11 is integrated, we can use FACS sorter to sort out those integrated cells because they are fluorescent. Here's the case with the knocking to the NAMI gene. We saw like 36% of the hexa23T cell line that becomes fluorescent. And we plate them. We can verify their fluorescence intensity coming from the nuclear envelope. So the whole procedure takes 1 day of experiment and a couple of days for the cell to grow up before we can do the sorting, and it costs about $100 to $150. That's very cost effective. And because our goal is to establish a method that we can really scale this method up to a large scale, I asked Manuel and Daiji and Sayaka both to try to pick their favorite target in genes and try to make a mini library. So the three of them spent 3 weeks. 1 week to pick the target gene and design the sequences. 1 week to wait for IDT to synthesize the DNA and send us the oligonucleotide. And another week later, we have the cell lines. We have a total of 12 ER proteins because of Sayaka's personal interest in ER morphology, 2 of the mitochondrial proteins, 5 of the genes associated with the cell skeleton and centrosome, and 8 of them associated with the nucleus. There's a clathrin for endocytosis, RAB11 for recycling endosomes, NAMP1 for lysosomes, and PKA because of other projects in the lab that's working on the protein kinase A. So out of the 48 targets they initially picked, 30 of them we immediately got FACS positive cells. And this 30, we typically have somewhere between 8 to 40% of the FACS positive cells, and that is a very efficient process. We can plot the whole cell fluorescence intensity measured by FACS against the expression level as measured by ribosome profiling. It's a pretty nice linear correlation. And for those ones that did not result in any FACS positive signal, those are the ones in brown color. You can see those are the ones that typically of lower expression level, kind of falling below the FACS detection limit. So if we set our FACS detection limit, that's about 27 RPKM, as say the threshold of whether we can get successful knocking or not. With this threshold, we will say that in HEP293 T cells, 30% of the genome is expressed above this level. So theoretically speaking, that we take the same approach, we should be able to do the knocking and cover about 30% of the entire human genome in HEP293 T cells. So that would be great, but it doesn't mean that for those ones that's falling below this threshold, we don't get integrated cells. If you look at the previous slide, we actually have 31 images. One of the images coming from NUC35 is a nuclear poring. It's too weak to be detected by FACS, but if we plate the mixed population, we can still see nuclear envelope signal coming from the fraction of the plated cells. So it's there. It's just the signal is not strong enough for our flow in cell sorter to detect it. Well, it also means if we can amplify the signal further, we will have a much better coverage of the whole genome. if we were to create knocking cell lines. Previously, what we've been doing is we have a target protein. We tag it with a copy of GFP, actually GFP11, to bring in the GFP1-10 so that it reconstitutes a full GFP. And our limitation of the signal is coming from the limitation of brightness of GFP. And that's kind of very difficult to improve because people have been working on GFP for so many years as to try to perfect it. So how to make it brighter? Well, initially, we tried the brute force way, and we cheated. And if one copy of GFP is not bright enough, how about having multiple copies of GFPs? But this is inspired by the Suntech methods coming out of Ron Ville's lab at UCSF. Basic idea is GFP11 is short enough, we should be able to arrange into 10 repeats. And then multiple copies of GFP11 will be able to bring in multiple copies of the GFP1-10, and that reconstitutes multiple copies of fluorescent GFP to amplify the signal. We tried this approach with 1x GFP11, 3x, 7x, and either with a 15 amino acid linker in between the repeats or with 5 amino acid linker, we saw a proportional amplification of the signals. That's nice. And here is our collaboration with the Wallace Marshall Lab trying to do single particle tracking using either fluorescent GFP or 7x GFP11 in the FT20 protein in the flagella and in the primary cilium. And you can see how much brighter when we have 7 copies of GFP11. And nice thing is that, at least for this case of FT20, either with GFP or 7x GFP11, we did not see a significant change in the transport speed of these particles. So it means that the function of this FT20 is not perturbed by having 7 copies of GFP dangling on it. So that will be a pretty easy way of amplifying the signal and improve our detection threshold. Well, the flip side is that now the DNA insert is larger and we cannot just directly synthesize the donor DNA. Emmanuel has worked out the protocol of using in vitro transcription and reverse transcription to make that single-strand donor DNA with multiple copies of GFP11. And we tried it on also the Namin gene. And here, about the same integration efficiency with 4x GFP11, we got 4 times the bright signal, which is seen both in a FACS histogram as well as in a fluorescent image. We also have to note that even with 7x GFP11, it's still much smaller than the GFP gene itself because GFP contains 11 beta strands plus an alpha helix, whereas GFP11 is just one of the beta strands. So it is a pretty effective way of amplifying the signal. And if we decrease our detection threshold by a factor of 4, we should be able to cover 60% of the genome in HEC203 T cells. That'd be nice. Still, it'd be nice if we can have a smaller tag. For this purpose, Siyu has looked at a newly engineered fluorescent protein, amnion green. And the full-length amnion green is about a factor of 2 to 3 as bright as full-length GFP. So that's great. And she has engineered a split version of that with multiple mutations to make it like self-complementing. And so here we can do amnion green 211, knocking to the 3 in genes, and they are much brighter. The other benefit is, well, we have not realized this issue until we really looked at data very carefully, that for whatever reason, cells expressed in GFP1-10 has higher fluorescent background compared to cells not expressing anything. And if you look at the orange curve, that's GFP1-10 expressing cell with a with a strong promoter. We can reduce the expression strength. If you look at the blue curve, the background's lower, but still there. We're not sure whether this is from some very weak residual fluorescence coming out of GFP1-10 or it's elevated autofluorescence coming out of GFP1-10. And it's a very weak fluorescent background, basically on the same order of magnitude compared to the autofluorescent background. So it never affects other applications, other powerful applications of the speed GFP, speed GFP11 system. But in our case, we're trying to detect a low expressing protein. This becomes an issue because the background is higher. In comparison, the amnion green is 1 to 10 is almost zero background, totally overlapped with the autofluorescence curve. So if you look at the histogram on the right, compare the GFP11 knocking versus amnion green 11 knocking for a low expressing protein, you can see the amnion green case is much better separated from the non-integrated cells because of lower background. So that is another advantage of the split amnion green system. Basically, for this reason, we pretty much all switched to amnion green in our own experiments nowadays. Well, now we can do very efficient knocking for green color fluorescent protein. Amnion green is slightly redshifted compared to GFP, but it's essentially still green colored. It would be nice to have a second color so that we can do double knocking and we can see protein-protein interactions or relative position of different proteins. When it comes to multicolor imaging with GFP, one of the classic targets, one of the classic fluorescent protein would be M cherry. Well, we've tried to split M cherry, but it ended up with no fluorescent signal at all. This is not surprising because the split GFP is not split EGFP. It's actually the Waldo lab started with engineering a super photo version of GFP for very efficient folding, and then further engineering after making the split to make it efficiently complementing. And it's no wonder if we split the ordinary amnion green, it doesn't work. Well, what to do? We tried the most powerful research engine, Google, and we type in super photo cherry. A paper did pop out. And it was also from the Waldo lab, and it was their work, but it was published in a fairly obscure crystallographer journal, which was all about a way to crystallize proteins. It's just the protein that crystallized is called super photo cherry. And later, I had a chat with the first author of this paper, saying that she was about to leave the lab, so she had to kind of publish this work that's been halfway through of trying to engineer the super photo version of cherry. Well, although this is kind of a half-baked product, we still tried to split this super photo cherry, and it worked. The very first trial, and we saw complementing fluorescence, although it's fairly weak. So this two-color image has been produced by a 4x repeat of SF cherry 11. Later, in my lab, tried to improve the brightness of super photo cherry split by tearing the two fragments with a 32 amino acid linker and then doing random mutagenesis, DNA shuffling, and directed evolution. She has derived the second generation of the split super photo cherry that is almost a factor of 8 as bright as the first generation. So these are the endogenous overexpressed case, and in the lower right corner, labeling of endogenous proteins using the SF cherry 2 11. Now we have the two colors. We can do double knocking, and that needs us to be able to do some very interesting things. For example, we've done double knocking to two of the ER proteins, 661B and R6AP1. And 661B has been widely used as a ER marker because it's a ER translocan protein, and its associated protein synthase supposedly has nothing to do with ER morphology, so supposedly it's to label all the ER. But to our surprise, when we're trying to enable these two proteins, we've seen at the very edge of the cell, the very peripheral yards, especially those dead end yard tubules, that has like fairly strong R6AP1, that's a ER tubule protein, neighboring, but very little 661B. And this is not an artifact of GFP11 versus Cherry 11 because we can swap these two labels when we saw pretty much the same thing at the very periphery of the cells. But more recently, Pedro Di Camini at Yale has published a paper, and they've also observed the same phenomenon, and they have some nice explanation of what might be going on, and that's associated with the function of R6AP1 protein. In any case, this is a very nice illustration that once we have the capability of enable endogenous proteins, we can, if we look at those endogenous proteins, we may have some nice surprising discoveries that people have not seen before because of the limitation of labeling. With the GFP11, amnion green 211, SF cherry 11, SF cherry 211, we actually think about this as a general approach to label proteins. And typically when people label proteins, they have this whole functional tag, and they put the tag on the target protein. This tag is big, that may perturb the function of proteins, or sometimes may make the labeling, make cloning difficult. In this case, in all these cases we've talked about so far, we're not tagging the target protein with the entire tag. We're just tagging the protein with a fragment of this of the tag. And when the larger fragment floating in the cytoplasm, and after the protein of interest is made with the fragment tag, the two pieces can come together that reconstitute the whole functional tag, and that has a lot of advantages. And in our case, when we talk about fluorescent proteins, there are other similar peptide large fragment interactions like spike tag, spike catcher, and we can in fact generalize this whole fragment tagging approach to many things. For example, we can use this to create photoactive low fluorescent tags. In this case, we merge the 6 mutations that converting M cherry into PA M cherry, and our mutations of converting M cherry into superphoto cherry 2. And that creates the photoactivatable superphoto cherry 1 to 10 fragment. It's share the same P11 fragment with SF cherry 211. So in this case, we can do endogenous protein labeling with a photoactive fluorescent protein, and then we can do superficial microscopy with a storm using this method here showing the storm image that nicely resolve the fine structural details of Catherine colipids. And similarly, we can do this thing with the spike tag, spike catcher. In this case, we actually used the AP11 fused in tandem, GFP11 fused in tandem with spike tags that's still small enough to fit in a 200 nucleotide donor DNA. And then the GFP11 as a selection marker, then leave the target protein with a spike tag handle. Later on, after fixation, we can do staining very efficiently with the spike catcher protein that forms a covalent isopeptide bond with the spike tag that bring in a 404 so that we can also do superficial microscopy storm with endogenous proteins labeled by tandem GFP11 spike tag. The labeling is super efficient because the interaction between spike tag and spike catcher is covalent, pretty much, well, completely irreversible. And we can use this tag for other purposes as well. Previously, when Jonathan and Aaron created that East library, it's actually 2 libraries. One GFP library for imaging, one TAP library for biochemical purification. Now, with the development of so many nice nanobodies against GFP, once we have a GFP11 knock-in library, we actually have a dual function library. GFP11 complement serves as a imaging tag, as well as a epitope tag for immunoprecipitation. In this case, we can do pull down on endogenous proteins. And here's proof of principle demonstration from Manuel. A number of complexes we have been showing that we can very cleanly pull down the other components of the same complex. So we are having a dual functional library that is not only useful for microscopy, but also useful for biochemical and even proteomics analysis. And with this library, with our ability to really scale things up as to have a potentially genomic cell library of the endogenous proteins tagged by the fragment tag, which is actually the task of Manuel Leonetti, who is now a group leader at the Cheongsa Biohub. And then hopefully one day we can really complete this pipeline for the entire genome and turn these nice illustrations by David Goodsell into real pictures. So that ends my presentation. I'd like to thank the people in my lab who participated in this project. I mentioned them during my talks. I would like to also thank our collaborators that's making all these things happening, as well as the funding agencies. And finally, every one of you for listening. Thank you.
Thank you, Dr. Huang, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Now let's get started. Our first question is, will the function of the target protein be perturbed when labeled by GFP11?
Well, that's a very important question, of course. With any fluorescent protein tagging methods, whether it's endogenous protein, whether it's overexpression, it's always important to make sure that the tag protein does not have its function perturbed. And supposedly in our case that although we are tagging the protein with just a short peptide GFP11, after complementation, it will be the same GFP as compared to when we tag the target protein with fluorescent GFP. So theoretically, you should not have more perturbation compared to fluorescent GFP tagging. In fact, for when we're trying to create our library, we pick the sites that people have already tagged those proteins with fluorescent GFPs to make sure that there's no expected function perturbation to the system. We did see strangely, in a couple cases that the target protein is tolerating GFP11 labeling, but not so much with fluorescent GFP enabling. Potentially, this is because that when the ribosomes are making the protein, and especially when when the GFP is started folding while the ribosome is making the protein, that may perturb the folding of the target protein potentially. But GFP11 is a short peptide that may have much less perturbation. But anyway, I think we always need to be careful when labeling protein with pretty much anything not to perturb the target function. And I would say that GFP11 is likely the one that has less perturbation compared to fluorescent GFP, if they are not having the same amount of perturbation.
And what is the affinity between the two fluorescent protein fragments?
Well, that's a difficult question. Because once the two fragments complemented, the whole protein is going to fold and the chromophore is going to mature. And from our experience, this process is pretty much irreversible, which means that if you want to biochemically measure the affinity between these two fragments, it's going to be very, very difficult. However, if we think this and treat this process as two steps, when first these two fluorescent protein can bind, and then they can fold, and then they can mature. There's still an affinity issue for the first process. Actually, in more careful characterization, that the GFP, for speed GFP, the two fragments has very, very high affinity. They come together very efficiently, and nearly 100% of the GFP level will be complemented by GFP110 and get into a fully mature GFP, fully mature GFP. This is not surprising given so much optimization that the Water Lab has done for this construct. For the newer split amnion green 1 to 10, 11 and split SF cherry 2, we realized that the complementation efficiency is not 1, meaning that there's still some affinity or binding dissociation issue between the two fragments before they fold and they irreversibly mature into a fully functional protein. It is one subject that we are looking into at this moment.
Okay, we have one more question. Will the expression of GFP 1 to 10 affect the cells?
That's a good question. GFP 1 to, in our experience, we've tried multiple cell lines expressing in GFP 1 to 10 or stable cell lines expressing GFP 1 to 10. We've even tried one iPS cell line generating one stable iPS cell line that is expressing GFP 1 to 10. GFP 1 to 10 seems to be pretty well tolerated in all these cases, but we do have to be careful that in GFP 1 to 10 it's not a completely folded protein. And so the cell may have certain unfolded protein response if there's too high an expression level of GFP 1 to 10, or there are too high the concentration of uncomplemented GFP 1 to 10 once it's in a cell. Now for the amnion green, we have not seen much difference in our limited experience. We have not seen much difference compared to GFP 1 to 10 in terms of perturbing the growth of the cell. But for SF cherry 1 to 10, we did have seen a little more cytotoxicity compared to GFP 1 to 10, as the cell may grow slower when there's too high an expression of SF cherry 1 to 10 in there. And these are our experience with the cell line. In general, they are pretty well tolerated by the cell line, but we do have to be careful not to overexert too much. And when it comes to like in animals and for organisms as compared to cell line, they may be not more sensitive. We have created GFP 1 to 10 Drosophila lines. The fly seems to be all okay with it. And I think this is a pretty interesting topic for further investigation to see how well this is tolerated by the cells, by various cell lines, by various organisms, and how well we can improve the whole system as to reduce their potential cytotoxicity.
All right, well, I would like to once again thank Dr. Huang for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.